Welcome to Chapter 3, which focuses on the structure and function of cells, the most fundamental unit of life. All living organisms, regarding of size or complexity, are built from cells, making this topic essential to everything we study in biology. The cell is the smallest unit that demonstrates all of the properties we associate with life, including metabolism, growth, responsiveness, and reproduction. As we move through this chapter, we'll examine how cell theory, explore differences between prokaryotic and eukaryotic cells, and discuss how structural features allow cells to perform specialized roles. We'll also explore how organelles divide labor within a cell, how the plasma membrane regulates movement and communication, and how metabolic pathways provide the energy that powers life's processes. By the end of this chapter, you should see how the activities inside even a single microscopic cell connect to the function of the entire human body. Cell theory is one of the most important unifying principles in biology. It is built upon the three essential concepts. First, all living organisms are made up of one or more cells, whether a single-celled bacterium or it's a multicellular organism like human beings. Second, the cell is the most basic unit of structure and function. It is the smallest entity that can exhibit all of the characteristics of life, including energy use, reproduction, and adaptation. No smaller structure, such as molecules or organelles, can be considered alive on its own. Third, the cell only arises from pre-existing cells. This means that life is continuous. Every cell today is descended from earlier cells, forming an unbroken chain back to the first cell billions of years ago. Before this theory was widely accepted, many people believed in the spontaneous generation, or the idea that life could appear from non-living matter. This rejection of that idea reshapes science and lays the foundation for modern microbiology, genetics, and medicine. Although all cells share a plasma membrane that encloses their internal environment, they are classified into two broad categories based on internal organization: prokaryotic and eukaryotic. Prokaryotic cells are more primitive, lacking the compartments or membrane-bound organelles that characterize eukaryotes. Their genetic material is found in the cytoplasm, and all of the cellular processes occur in one undivided space. By contrast, eukaryotic cells have a nucleus which houses DNA and a variety of organelles, each performing specialized tasks. This separation of function is critical for efficiency, allowing spontaneous processes like energy production, protein synthesis, and waste management. The plasma membrane acts as a selective barrier in both cell types, regulating movement of substances and communication with the environment. This fundamental division between prokaryotes and eukaryotes is one of biology's most important distinctions, shaping everything from evolution to the complexity of organisms. Eukaryotic cells are the type found in humans, animals, plants, fungi, and protists. Their hallmark feature is compartmentalization. The nucleus serves as the command center of the cell, housing DNA and directing cellular activity. Surrounding it is the cytoplasm, which includes the cytosol, a gel-like fluid that supports and suspends the organelles. Each organelle has a distinct role that contributes to the overall function of the cell. Mitochondria supply energy in the form of ATP, while the endoplasmic reticulum and Golgi apparatus manufacture, modify, and distribute proteins and lipids. Lysosomes recycle cellular debris, and peroxisomes detoxify waste. This division of labor allows for remarkable efficiency and complexity, permitting eukaryotic cells to support multicellular life. Their internal organization provides the basis for specialization. For example, muscle cells packed with mitochondria for energy, or liver cells rich in smooth ER for detoxification. Understanding organelles help us link structure to function across tissues and systems. Prokaryotic cells, which include all members of the domain bacteria and archaea, are structurally simpler but remarkably successful. Unlike eukaryotes, they lack a nucleus, and their DNA exists in a single circular chromosome located in the cytoplasm in the area called the nucleoid. They lack membrane-bound organelles, so all processes like protein synthesis, energy metabolism, and waste handling occurs in the same space. Many prokaryotes have a rigid cell wall that provides the structural support and protection, and some possess additional features like capsules or flagella that enhance survival. Prokaryotes are generally smaller than eukaryotes, but this simplicity gives them advantages. They reproduce quickly, adapt rapidly to environmental changes, and colonize every habitat on Earth, from beneficial gut bacteria that aid in digestion to harmful pathogens that cause disease. Prokaryotes play essential roles in ecosystems and human health, demonstrating that complexity is not only path to success in biology. Although all eukaryotic cells share basic structures, they are not identical. Each type of cell is specialized to carry out particular roles, and its shape and organelles reflect that specialization. For example, muscle cells are packed with mitochondria because constant energy production is necessary for contraction. Nerve cells, in contrast, are elongated and thin, allowing them to transmit electrical signals over long distances. Red blood cells are small and disc-shaped and maximize surface area for oxygen transport. These structural differences demonstrate an important theme in biology. Form follows function. All cells must take in raw materials, remove waste, and synthesize macromolecules, grow and reproduce, but how they do so depends on their design. By examining structure we can often predict function. This principle helps us understand not just healthy cells, but also what happens when structure is altered, as in cancer or genetic diseases, where abnormal form leads to abnormal function. Cells remain microscopic because small size maximizes efficiency. As a cell grows larger, its volume increases faster and its surface area, which makes transporting nutrients in and waste products out more difficult. Small cells have a high surface area to volume ratio, ensuring enough plasma membrane to meet metabolic demands. For example, oxygen must diffuse into the cell and carbon dioxide must diffuse out. If the cell is too large, diffusion becomes inefficient and the cell cannot survive. To solve this, cells either remain small or adapt strategies to increase surface area, such as having folds, extensions, or elongated shapes. Some cells, like intestinal epithelial cells, have microvilli that dramatically expand their surface area to maximize absorption. This principle of size and limitation helps explain why multicellular organisms evolved. Many small cells working together can form tissues and organs that function more efficiently than one giant cell ever could. Because cells are too small to see with the naked eye, microscopes are an essential tool for studying them. The light microscope uses visible light and glass lenses to magnify objects up to 1,000 times, making it ideal for viewing living specimens such as cells in motion. However, its resolution is limited, meaning they cannot see fine details inside the cell. For that, electron microscopes are used. The transmission electron microscope provides magnification up to 100 times or more that reveal internal structures such as organelles, membranes, and even large molecules. The scanning electron microscopes also reaches high magnification but provides a detailed three-dimensional view of the cell surface, showing texture and shape. Together, these instruments revolutionize our understanding of cell biology. Microscopy not only allows us to observe structures but also connects structures to function, helping us investigate disease, development, and cellular processes in real time. Cells are complex systems with specialized internal structures that perform distinct tasks. The nucleus functions as the information center containing DNA that directs all cellular activity. Ribosomes build proteins, the workhorses of the cell. The endoplasmic reticulum acts as the manufacturing center, while the Golgi apparatus refines, packages, and ships products to their destination. Vesicles serve as the storage and transport containers, moving materials within and outside of the cell. Mitochondria provide energy by generating ATP, the currency cells use to process and power work. These structures highlight the concept of compartmentalization. Different processes occur in specific organelles, preventing interface and increasing efficiency. This divisional labor allows eukaryotic cells to perform complex functions that prokaryotes cannot. When many organelles malfunction, the entire cell suffers, reminding us that the health of the cellular level is directly tied to the health of the whole organism. The nucleus is often referred to as the control center of the cell because it houses the genetic material, DNA, which contains the instructions for every protein and process in the organism. Structurally, the nucleus is surrounded by a double-layered nuclear membrane that provides protection while allowing regulation exchange within the cytoplasm. Nuclear pores are specialized channels that permit RNA and proteins to pass in and out, ensuring communication between the nucleus and the rest of the cell. Inside, DNA is organized into chromosomes which carry hereditary information. The nucleolus, which is a dense structure within the nucleus, is the site of ribosome component synthesis, preparing the machinery for protein production. By directing gene expression, the nucleus ultimately controls growth, metabolism, and reproduction. Damage to the nuclear DNA, as seen with mutations, can disrupt these instructions and lead to diseases such as cancer. Thus, the nucleus is both the library of information and the director of cellular activity. Ribosomes are small but essential structures where proteins are made. They are composed of ribosomal RNA and proteins that assemble amino acids into polypeptides according to the instructions carried out by the messenger RNA. Ribosomes can be free in the cytoplasm or bound to the surface of the rough endoplasmic reticulum. Free ribosomes produce proteins that function within the cell itself, while bound ribosomes make proteins that are designed and destined for secretion, insertion into membranes, or delivery to organelles. This division ensures that the proteins reach the correct destination which is critical for maintaining cell function. Although ribosomes are not surrounded by a membrane, they are considered organelles because of their specialized function. Their importance cannot be overstated. Proteins carry out structural roles, catalyze reactions as enzymes, and serve as a signaling molecule. Without ribosomes, cells would not produce the proteins necessary for survival, growth, repair, or communication. The endoplasmic reticulum is a highly folded network of membranes that serve as the cell's manufacturing center. It comes in two forms, rough and smooth endoplasmic reticulum. Rough ER is studded with ribosomes, giving it a bumpy appearance. Those ribosomes manufacture proteins, many of which are modified in the ER before being sent out to the next destination, especially those destined to be exported or incorporated into membranes. Smooth ER, which lacks ribosomes, is responsible for lipid and hormone synthesis as well as detoxification of harmful substances. It also plays a role in packing proteins and lipids for transport to the Golgi apparatus. The extensive folding of the ER increases surface area, maximizing efficiency. Cell specialization in protein secretion, such as pancreatic cells, have abundant rough ER, while cells involved in lipid metabolism or detoxification, like liver cells, are rich in smooth ER. Together, both forms of ER contribute to the cell's ability to grow and adapt and maintain homeostasis. The Golgi apparatus acts as the cell's packaging and distribution center. It receives products from the endoplasmic reticulum, refines them, and directs them to their final destination. Structurally, the Golgi consists of stacked, flattened membranes, often compared to a series of pancakes. Inside, proteins and lipids undergo modification, such as in the addition of carbohydrate groups, which serve as address labels to tell the cell where to send them. Once processed, these products are packed into vesicles. Some vesicles deliver materials to other organelles, while others move to the plasma membrane for secretion outside of the cell. This system ensures accuracy, like the post office sorting and delivering mail. The Golgi is particularly prominent in cells that secrete large quantities of substances like digestive enzymes, mucus, or hormones. Malfunctions in the Golgi processing can have serious effects, leading to misdirected or defective proteins that contribute to disease, including certain congenital disorders. Vesicles are small membrane-bound sacs that serve as storage and transport units within the cell. There are several specialized types. Secretory vesicles contain products that need to be exported, such as hormones or neurotransmitters, and release their contents through exocytosis. Endocytotic vesicles form when the plasma membrane engulfs substances from the external environment, allowing materials like nutrients or signaling molecules to enter the cell. Lysosomes contain digestive enzymes that break down old organelles, cellular debris, and macromolecules, essentially serving as the cell's recycling center. Peroxisomes contain enzymes that neutralize toxins and break down harmful byproducts of metabolism. Vesicles keep substances separated from the rest of the cytoplasm until they are needed or eliminated, ensuring that the cell remains organized and protected. By coordinating storage, delivery, and waste disposal, vesicles contributes to the cell's overall efficiency and survival, much like a delivery and sanitation system in a busy city. Mitochondria are the powerhouse of the cell, generating energy in the form of ATP through cellular respiration. They are unique in that they have a double membrane, which is an outer membrane that defines the boundary, and a highly folded inner membrane that forms the cristae, which greatly increase the surface area for energy production. The space inside the inner membrane, called the matrix, contains enzymes needed for the citric acid cycle, while the intermembrane space plays a role in establishing the proton gradient necessary for ATP synthesis. Mitochondria use oxygen and release carbon dioxide as a byproduct as they break down glucose and other fuel molecules. The number of mitochondria in a cell varies depending on its energy demands. Muscle cells contain thousands, while less active cells may have fewer. Remarkably, mitochondria contain their own DNA and can replicate independently, suggesting an evolutionary origin free from living bacteria. Their role is central. Without mitochondria producing ATP, most cellular processes would quickly come to a halt. In addition to mitochondria producing ATP from glucose, they also rely on stored energy reserves in the form of fat and glycogen. These molecules are not enclosed by membranes but are stored directly in the cytoplasm. Fat, stored as triglyceride, represents a long-term energy storage. Fat cells, or adipocytes, can hold large amounts to provide a reserve that can sustain the body for extended periods. Glycogen, by contrast, is a carbohydrate storage molecule found mainly in the liver and the muscle cells. It serves as short-term energy storage and can be quickly broken down to glucose when immediate energy is needed. During exercise, muscle glycogen is used to fuel contractions, while liver glycogen helps maintain blood glucose levels. The body manages these reserves carefully, using glycogen first to quickly burst for energy and turning to fat when long-term or large energy demands occur. This balance ensures that survival during both everyday activities and times of fasting or stress. Cells are not just static sacs of fluid, they contain structural elements that provide support and enable movement. The cytoskeleton forms an internal scaffolding that maintains the cell shape and organization. Without it, cells would collapse or lose their form. The cytoskeleton also anchors organelles and guides intercellular transport, acting like railroad tracks for vesicles. In addition to internal support, cells can move or manipulate their environment using specialized structures. Cilia and flagella are extensions that beat or whip to produce movement, either moving the cell itself or moving materials across the surface. Centrioles, short cylindrical structures located near the nucleus, play a critical role in division by organizing the spindle fibers that separate chromosomes. Together, these elements ensure cells can adapt, change shape, divide, and interact with their surroundings, which is a vital process that, like tissue development, immune response, and reproduction. The cytoskeleton is composed of protein filaments that provide mechanical support and dynamic organization to the cell. Its two main components are microtubules, which are hollow tubes of proteins, and microfilaments, which are thin, solid fibers. Microtubules give rigidity and help maintain the cell's overall structure, while microfilaments are involved in the movement and shape changes. The cytoskeleton acts as the poles of a tent, supporting the membranes while also allowing flexibility. Beyond structural support, it provides tracks for the movement of organelles, vesicles, and even chromosomes during cell division. Motor proteins walk along these tracks, carrying cargo to different locations. The cytoskeleton is not static. It's constant assembly and dissembles, allowing cells to exchange shape, migrate, and to respond to signals. This dynamic system is essential for processes like wound healing, immune defense, and embryonic development. Cilia and flagella are cellular structures specialized for movement. Cilia are short and numerous, often covering the surfaces of cells. They beat in coordinated waves to move fluid or particles across the surface, such as clearing mucus from the respiratory tract. Flagella, in contrast, are longer and usually appear singly, propelling cells forward with a whip-like motion. Human sperm cells rely on flagellum for motility. Despite their differences in appearance, both cilia and flagella share a similar internal structure based on the microtubules arranged in a 9+2 pattern. Centrioles, which are located near the nucleus, play an important role in organizing these structures and especially critical during cell division. Together, the motility structures not only enable movement but also help the cells interact with their environment. Without cilia, for example, the respiratory system could not effectively clear debris, and without flagella, sperm could not reach the egg, demonstrating their vital roles in health and reproduction. The plasma membrane is a thin boundary that surrounds every cell, separating the internal environment from the outside world. It is selectively permeable, meaning it allows some substances to cross while blocking others, thus maintaining homeostasis. This selective transport ensures that essential nutrients like glucose and amino acid enter while wastes are removed. The membrane also enables cells to sense and respond to their environment through receptors that receive chemical signals. In this way, the plasma membrane is not just a barrier but also a communication interface. For example, hormones binding to receptors on the membrane can trigger changes inside the cell. Its role is critical for survival. Without it, cells could not regulate their internal composition, maintain energy balance, or coordinate with other cells in the tissue. Think of the plasma membrane as both a security gate and an information hub, controlling access and relaying messages that guide cellular behavior. The plasma membrane is built from a lipid bilayer that gives it both structure and flexibility. The bilayer is composed mainly of phospholipids, each with a hydrophilic head and a hydrophobic tail. The head faces outwards towards the watery environment, while the tail meets the middle, forming a hydrophobic core. This arrangement creates a selective barrier. Cholesterol molecules are interspread throughout, adding mechanical strength and stability, preventing the membranes from becoming too rigid or too fluid. Proteins are also embedded, serving as channels, carriers, receptors, and enzymes. Because components are not fixed in a place, the membrane is described as a fluid mosaic, where lipids and proteins drift laterally. This fluidity allows the cell to adapt and change, repair itself, and form dynamic structures like vesicles. Understanding the bilayer is crucial since its properties explain how nutrients enter, how waste leaves, and how cells receive information, fundamental processes that help life running at the cellular level. Molecules cross the plasma membrane in several ways, and the method depends on its size, polarity, and energy requirement. Passive transport requires no energy and relies on natural movement down a concentration gradient. Diffusion allows small, nonpolar molecules like oxygen or carbon dioxide to slip through the lipid bilayer. Osmosis is the diffusion of water across the membrane, moving towards higher cellular concentrations to balance fluid. Facilitated diffusion uses proteins that provide a pathway for longer or charged molecules, such as glucose or ions, but still does not require energy. In contrast, active transport moves substances against the concentration gradient from low to high concentration. Active transport moves substances along the concentration gradient and requires ATP. This allows cells to accumulate nutrients or expel waste regardless of concentration differences. For moving very large molecules or bulk materials, cells use vesicles. Endocytosis brings substances in, while exocytosis expels products or wastes. Together, these processes ensure cells maintain homeostasis and interact efficiently and effectively with the environment. Passive transport moves substances across the membrane without energy expenditure. The most basic form is diffusion, the movement of molecules from an area of high concentration to low concentration, down the concentration gradient. For example, oxygen diffuses into the cell where it is at lower concentration, while carbon dioxide diffuses out. When concentration equalizes, equilibrium is reached. However, diffusion is only effective over short distances, which is why cells remain small. Osmosis is a specialized form of diffusion involving water movement through a selectively permeable membrane. Water moves from areas of low solute concentration to areas of high solute concentration, effectively balancing solute levels on both sides. Osmotic pressure is the force needed to counteract this flow. Both diffusion and osmosis are critical for maintaining homeostasis, allowing nutrients, gases, and waters to move naturally without consuming ATP, ensuring the cell can focus energy on other demanding processes. Most molecules cross the membrane by passive transport, meaning they move with, not against, the concentration gradient. This process requires no cellular energy. There are three main mechanisms: diffusion directly through the lipid bilayer, diffusion through protein channels, and facilitated transport. Small nonpolar molecules like oxygen, carbon dioxide, and urea slip directly through the lipid bilayer. Larger or charged molecules require assistance. Protein channels provide a passageway for ions or water molecules, either remaining open or opening under specific conditions. Facilitated transport uses carrier proteins that undergo shape changes when binding to specific substances like glucose or amino acids to move them across the membrane. In each case, movement occurs from high to low concentration. Passive transport is efficient and essential for survival, ensuring continuous exchange of substances that support metabolism and other cellular activities without depleting the cell's limited energy resources. The simplest way for a molecule to cross the plasma membrane is by diffusion directly through the lipid bilayer. Because the bilayer's core is hydrophobic, only small nonpolar lipid-soluble molecules can pass easily. Examples include oxygen, which diffuses into the cell for respiration, carbon dioxide, which diffuses out as waste, and urea, a byproduct of the protein's metabolism. This direct diffusion is efficient and requires no protein or energy, but it is limited to molecules that can dissolve in lipids. Large molecules, ions, or polar substances cannot cross this way, since they are repelled by hydrophobic core. Despite its limitations, direct diffusion is critical for processes like gas exchange in the lungs and tissues. Without it, oxygen cannot reach the mitochondria to fuel ATP production, and carbon dioxide would accumulate in toxic levels. Protein channels expand the capability of the plasma membrane by allowing molecules that cannot diffuse directly through the lipid bilayer to enter or exit the cell. These channels are highly selective, permitting passage based on size, shape, or electrical charge. Some channels are always open, providing continuous access for molecules like water, while others are gated and open only in response to signals such as changes in voltage or binding of a molecule. This selectivity ensures precise regulation of cellular environments. For example, water often moves through a specialized protein channel called aquaporins, enabling rapid osmosis. Ions such as sodium, potassium, and chloride also rely on channels to move in and out critical for processes like nerve signaling and muscle contraction. Because movement still occurs down the concentration gradient, no energy is required. These channels illustrate how proteins embedded in the membrane expand its function beyond its simple barriers in a finely tuned gateway. Facilitated transport, also known as facilitated diffusion, helps larger or polar molecules cross the plasma membrane with the assistance of transport proteins. Unlike simple diffusion, these molecules cannot move through the lipid bilayer on their own. Instead, they bind to a specific transporter protein embedded in the membrane. The protein changes shape and shuttles the molecule across to the other side, always moving down its concentration gradient. Because the movement does not require energy, this is still considered passive transport. Examples include glucose and amino acids, both vital for cell metabolism. Transport proteins are highly selective. Each is designed for only one or few molecules, ensuring precise regulation of what enters or exits the cell. This process demonstrates the balance between efficiency and control. The cell conserves energy while maintaining the ability to regulate nutrient uptake. Without facilitated transport, cells would struggle to acquire the large building blocks required for growth and survival. Active transport is the opposite of passive because it moves substances against their concentration gradient, from areas of low concentration to high concentration. This requires energy, usually in the form of ATP, and specialized membrane proteins, often referred to as pumps. A classic example is the sodium-potassium pump, which maintains ion balance critical for nerve impulse and muscle contraction. This pump expels sodium ions from the cell while bringing in potassium ions, both against their gradients. By doing so, the pump not only regulates ion concentration but also contributes to maintain proper cell volume and electrical charge. Active transport is essential when cells need to accumulate nutrients even if they are scarce outside or expel waste even if its concentrations are higher inside. Some materials are too large to move across the plasma membrane through channels or transport proteins. In these cases, cells use bulk transport processes known as endocytosis or exocytosis. In endocytosis, the cell engulfs materials by folding in parts of its plasma membrane, forming a vesicle that carries the substance inside. This is how cells take in nutrients, fluid, or even large particles. Endocytosis can be selective when receptor proteins capture specific molecules, such as insulin binding, before being internalized. Exocytosis is the reverse process. Vesicles contain substances fused with the plasma membrane and release their contents outside of the cell. This mechanism is critical for secretion of hormones, neurotransmitters, and digestive enzymes. Both processes require energy and allows the cell to communicate, defend themselves, and maintain balance. The plasma membrane is not a physical barrier. It is also a communication hub. Cells rely on the receptor proteins embedded in the membrane to detect and respond to signals from the external environment. These receptors have specific binding sites for molecules such as hormones or neurotransmitters. When signaling molecules bind, the receptor undergoes a conformational change that triggers a cascade of events inside the cell. For example, when insulin binds to its receptor, it stimulates glucose transport into the cell. Different cell types possess different receptors, giving them unique sensitivities and function. This communication is essential for coordinating activities across tissues and organs. Without it, cells would act in isolation, unable to respond to environmental changes or maintain homeostasis. The sodium potassium pump is one of the best examples of active transport and is vital for maintaining both ion balance and cell volume. This pump uses ATP to move 3 sodium ions out of the cell while bringing in 2 potassium ions, both against their concentration gradients. By its doing so, the pump prevents sodium buildup, which would cause water to flow in by osmosis and potentially swelling or bursting the cell. At the same time, it ensures a high concentration of potassium inside, which is essential for nerve impulse, muscle contraction, and enzyme function. The pump operates continuously, consuming a large portion of the cell's ATP supply, underscoring its importance. If pumping slows, sodium accumulates, water flows, and the cell enlarges. If pumping speeds up, excess sodium is expelled and the volume decreases. This delicate regulation ensures that the cell remains stable, energized, and ready to perform their specialized roles in the body. Tonicity refers to the relative concentration of solutes inside versus outside of the cell, and it has a direct effect on cell volume. In an isotonic environment, solute concentrations are equal inside and outside of the cell, meaning water movement in and out is balanced. This allows cells to maintain their normal size and function. In the human body, most extracellular fluid is carefully regulated to maintain isotonic with intercellular fluid, ensuring stable conditions for metabolism and communication. If this balance is disrupted, cells can shrink or swell, leading to impaired function or even death. For example, intravenous fluid administered in medicine is isotonic solution to avoid harming red blood cells. This concept highlights how closely regulated the body's internal environment must be. When extracellular fluid is not isotonic, cells experience stress. In a hypertonic solution, the solute concentration outside of the cell is higher than inside. Water leaves the cell by osmosis, causing the cell to shrink, a process called crenation in red blood cells. In a hypotonic solution, the solute concentration outside the cell is lower than inside. Water rushes in and the cell swells, which may lead to lysis or bursting. These effects demonstrate how sensitive cells are to their environment. For example, drinking seawater exposes cells to hypertonic conditions, leading to dehydration. Conversely, excess water intake without electrolytes can cause hypotonic stress, endangering brain cells. To prevent such problems, organisms regulate extracellular fluid through mechanisms such as kidney function and hormone activity. Metabolism refers to the sum of all chemical reactions in an organism. These reactions transform matter and energy in ways that allow life to persist. Thousands of reactions occur spontaneously within a cell. often organized into pathways that are linear, branched, or cyclic. Together, they sustain processes like growth, repair and communication, and energy balance. Metabolism has two broad categories, anabolism and catabolism. Anabolism builds up complex molecules like proteins and nucleic acids from smaller subunits requiring energy input. Catabolism breaks down large molecules such as glucose into smaller products releasing energy the cell can capture in ATP. Both processes are interconnected. The energy released by catabolism fuels anabolic reactions. This constant cycling of building and breaking maintains life. When metabolism becomes imbalanced, diseases may result such as obesity, diabetes, or inherited metabolic disorders. Anabolism and catabolism are complementary aspects of metabolism. Anabolism refers to constructive processes in which small molecules are joined to build larger, more complex ones. These reactions require energy, often provided by ATP. For example, cells link amino acids together to form proteins, or glucose molecules to form glycogen. Catabolism, by contrast, is destructive, breaking down large molecules into smaller ones while releasing energy. A prime example is cellular respiration, where glucose is degraded into carbon dioxide and water by generating ATP. These processes are tightly linked. ATP generated by catabolism powers anabolic reactions, creating a cycle of energy, use, and renewal. This balance ensures that the cells have the resources to grow, divide, and respond to their environment. Every chemical reaction in a cell requires an enzyme to proceed efficiently. Enzymes are biological catalysts that lower the activation energy, allowing reactions to occur at rates compatible with life. Without enzymes, most reactions would be too slow to sustain cellular functions. Metabolic activities also demand a constant supply of energy. Cells use ATP as their energy currency to drive chemicals and drive critical tasks such as building macromolecules, transporting substances against gradients, and powering movement like muscle contraction. ATP releases energy when one of its phosphate bonds is broken, forming ADP, an inorganic phosphate. The cell then generates ATP through catabolic processes, especially cellular respiration. This constant cycle of ATP breakdown and renewal powers nearly everything a cell does. If an enzyme malfunctions, as in certain genetic disorders, or if ATP production falters, as in mitochondrial diseases, the cell cannot maintain normal function. Thus, enzymes and ATP together form the backbone of metabolic life. Glucose is the primary fluid molecule of most cells, and its breakdown supplies the majority of ATP. A single glucose molecule can yield up to 36 ATP through cellular respiration, making it highly efficient. ATP acts as a convenient, immediately usable energy source compared to glucose, which must first be processed. When glucose is scarce, other molecules such as fats and proteins can be catabolized, but glucose remains the body's preferred source because it can be mobilized quickly and metabolized efficiently. Think of glucose as a large bill that must be exchanged for smaller denominations of ATP, which the cell can then spend on specific tasks like muscle contraction, transport, and biosynthesis. Cellular respiration is the process by which glucose is broken down in the presence of oxygen to generate ATP. It involves four major stages: glycolysis, the preparatory step, the citric acid cycle, and the electron transport chain with oxidative phosphorylation. Each stage takes place in a specific location and contributes to the overall yield of ATP. Glycolysis occurs in the cytoplasm and breaks down glucose into pyruvate molecules, producing a small amount of ATP and electron carriers. The preparatory step converts pyruvate to acetyl coenzyme A inside the mitochondria. The citric acid cycle in the mitochondrial matrix extracts high energy electrons and produces more ATP and carbon dioxide as waste. Finally, the electron transport chain located in the inner mitochondrial membrane produces the majority of ATP through oxidative phosphorylation. Together, these stages illustrates the intricate coordination of enzymes and organelles needed to transport glucose into usable energy efficiently. Glycolysis is the first step of cellular respiration and occurs in the cytoplasm. It involves a series of 10 enzyme-catalyzed reactions that break down one glucose molecule into two pyruvate molecules. This process requires an initial investment of 2 ATP, but yields 4 ATP, producing a net gain of 2 ATP. Glycolysis also generates high energy electron carriers. NAD plus picks up electrons and hydrogen ions to form NADH, which we will later contribute to ATP production in the mitochondria. Although glycolysis is relatively inefficient compared to later stages, it has two key advantages. First, it does not require oxygen, so it can proceed under anaerobic conditions. Second, it provides pyruvate, the essential fuel for further aerobic metabolism. After glycolysis, the two pyruvate molecules enter the mitochondria, where they undergo the preparatory step. Each pyruvate is converted into an acetyl group, releasing carbon dioxide as a waste product. This is one reason why we exhale carbon dioxide. During this conversion, electrons and hydrogen ions are transferred to NAD+, forming NADH, another important electron carrier. The acetyl group then binds to acetyl coenzyme A, producing acetyl coenzyme A, which is the entry molecule for the citric acid cycle. Although no ATP is directly generated during this step, it is critical because it links glycolysis, which occurs in the cytoplasm, to the aerobic process inside mitochondria. This preparatory step also ensures that high energy electrons are harvested early, maximizing efficiency. The citric acid cycle, also called the Krebs cycle, occurs in the mitochondrial matrix and is a cyclic series of 8 enzymes catalyze reactions. Acetyl coenzyme A enters the cycle by donating its acetyl group to oxaloacetate, forming citric acid. As the cycle progresses, citric acid is broken down, releasing carbon dioxide as waste, and regenerates oxaloacetate to continue the process. Along the way, high-energy electrons are extracted and transferred to the electron carriers NADH and FADH2, which transfers them to the electron transport chain. Each turn of the cycle produces one ATP directly, and since 2 acetyl coenzyme A molecules enter per FADH per glucose, the cycle yields 2 ATP total. More importantly, it generates abundant NADH and FADH2, which are the main sources of energy for the final stage of respiration. The electron transport chain is the final stage of cellular respiration and the site of most ATP production. It is located in the inner mitochondrial membrane where proteins and electron carriers are embedded. NADH and FADH2 generated in the earlier steps donate high-energy electrons to the chain. As electrons pass from one carrier to the next, they release energy used to pump protons into the intermembrane space, creating a concentration gradient. This gradient represents stored potential energy, like water behind a dam. The final electron acceptor is oxygen, which combines with electrons and protons to form water, highlighting the oxygen is essential for life. Without oxygen, the chain stops and ATP production halts. The process of coupling electron transfer with ATP generates is called oxidative phosphorylation. This electron transport efficiency allows one glucose molecule to yield about 30 ATP here, understanding its central role in cellular energy production. Oxidative phosphorylation refers specifically to the process by which ATP synthase uses the proton gradient created by the electron transport chain to generate ATP. As electrons move down the chain, energy pumps hydrogen ions from the matrix into the intermembranous space, producing a steep electrochemical gradient. Protons naturally want to flow back into the matrix, and ATP synthase provides the channels. As hydrogen ions pass through the enzyme harnesses their kinetic energy to phosphorylate ADP, forming ATP. This process is extraordinarily efficient and accounts for the bulk of ATP generated during respiration. The dependence on the oxygen as the final electron acceptor ensures a continuous flow of electrons, making oxidative phosphorylation aerobic. When oxygen is unavailable, the system stalls and the cells must rely on less efficient pathways like glycolysis. This mechanism highlights the elegant design of cellular respiration, where the movement of tiny particles is converted into energy that powers all of life's processes. Cellular respiration involves over 20 enzyme-catalyzed reactions that work in sequence to gradually release the energy stored in glucose. If all this energy were released at once, it would be wasted as heat. Instead, cells capture it in manageable amounts, storing it in the bonds of ATP. From one glucose molecule, the theoretical net yield is about 36 ATP, though the exact number may vary. Along the way, oxygen is consumed and carbon dioxide is released as waste. This process shows how cells act like power plants, taking in fuel and oxygen, producing usable energy, and expelling waste gases. The efficiency of respiration is remarkable compared to simple combustion, which releases energy as heat but cannot be captured for it to work. Although glucose is the body's primary energy source, fats and proteins also provide fuel when needed. Glycogen, stored in the liver and muscle, represents about 1% of the total reserves and can be rapidly converted back to glucose for immediate energy. Fats, however, make up nearly 78% of the reserves and are highly efficient, yielding more than twice the energy of carbohydrates, gram per gram. Proteins provide about 21% of the reserves, but they still are not normally used for energy since they serve structural and enzymatic roles. When required, proteins are broken down into amino acids, which can then enter the citric acid cycle at various points. Fats and proteins provide metabolic flexibility, ensuring that the body can continuously generate ATP when glucose is limited. Fats are essentially important energy sources because they store more than twice the energy of carbohydrates. When fats are broken down, triglycerides are split into glycerol and fatty acids. Glycerols can be converted into glucose in the liver or processed into pyruvate to enter the citric acid cycle. Fatty acids are also metabolized differently. They are broken down into two carbon units to form acetyl coenzyme A, which then enters the citric acid cycle directly. This process, called beta oxidation, generates large amounts of NADH and FADH2, feeding into the electron transport chain to produce even more ATP. Fat is the body's preferred long-term energy storage because it is compact and energy dense. For example, adipose tissue allows animals, including humans, to survive long periods without food. However, mobilizing fats takes longer than using glycogen, which is why the body uses glycogen for quick bursts of activity and fat for potential energy demands like endurance exercises or fasting. Although proteins can be used as fuel, this is not their primary role in the body. Proteins can be broken down into amino acids during digestion and metabolism. To use amino acids for energy, the amine group, the NH2, must first be removed through deamination. This produces ammonia, which is toxic, so the liver converts it to urea and then is excreted in the urine. The remaining carbon backbone of the amine group can be converted to pyruvate, acetyl coenzyme A, or other intermediates of the citric acid cycle, where it contributes ATP production. However, the body prefers to preserve proteins for critical roles like enzyme function, structural support, and cell signaling. Protein catabolism increases during starvation or extreme stress, when glycogen and fat stores are depleted. At such times, muscle tissue may be broken down to supply amino acids for energy. When oxygen is unavailable, aerobic respiration cannot proceed, but cells can still generate limited ATP through anaerobic pathways. Glycolysis continues to break down glucose into pyruvate, yielding to ATP per molecule. Without oxygen, however, pyruvate cannot enter the citric acid cycle and instead is converted into lactic acid. In muscles, this buildup can cause fatigue and the burning sensation experienced during intense exercise. Although anaerobic respiration is far less efficient than aerobic pathways, it provides a short-term solution that allows survival in low oxygen conditions. Once oxygen becomes available, again, lactic acid is converted back into pyruvate and metabolized aerobically, clearing the buildup. Anaerobic pathways also use the certain microorganisms that thrive in oxygen-poor environments. For humans, they are a temporary backup system. This process highlights the adaptability of metabolism, ensuring ATP is available when conditions are not ideal, although the cost of efficiency and endurance